Swayam Prabha. Digital India. Educated India.
Previous lectures we have discussed about the general principle of electrophoresis. We have discussed about the factors which affects a successful electrophoretic run like electrophoretic mobility, heating, that is Joule heating, and the electroosmotic flow. Also, if you could recall, we have discussed about horizontal and vertical gel electrophoresis. As we were discussing, the two most widely used matrices are agarose gels and the polyacrylamide gels. Agarose gels are mostly utilized for separating nucleic acids, whereas polyacrylamide gels are mostly utilized for protein separation. Polyacrylamide gels could be utilized for DNA separation, that is of very small DNA fragments, and likewise proteins could be utilized for the agarose gels could be utilized for not really protein separation, but where you can have certain analysis like say immunoelectrophoresis or isoelectric focusing, where sieving effect is not required actually. So, there protein could be analyzed. We will continue our discussion on electrophoresis and extend our discussion from where we have left. So, if you could recall, we left at the vertical and horizontal gel electrophoresis. Now, there are different kinds of buffer systems which are utilized in electrophoresis. And buffer systems, when we are talking about is when you cast the gel or when you polymerize the gel, you polymerize them in a particular buffer. Now, this electrolyte buffer plays a very important role in electrophoresis separation or in experiments, and it greatly affects the separation and resolution. So, electrolyte buffer is very important and greatly affects the separation and resolution of a particular analyte. Now, proteins differ quite a lot in their sensitivity to pH, ionic strength, types of ions, and as we have discussed that proteins, the charge on proteins will depend on what pH buffer you are taking, and that will depend on what is the pi value of that protein, as proteins are amphoteric molecules and they can carry either positive or negative charge as per the pH and their pi values actually. Now, in electrophoresis there are two types of buffer systems. One is called continuous buffer system and another is discontinuous buffer system. It can also be called as multiphasic buffer system. So, continuous buffer system is a single buffer system, that where you are utilizing single buffer, whereas in discontinuous system you will have more than one kind of buffer or maybe two buffers or three buffers. Now, for nearly all electrophoresis of concerning nucleic acids, mostly continuous systems are utilized either in agarose gels or polyacrylamide gels, whereas the electrophoresis of proteins is mostly done on a discontinuous system. So, there is a difference in here. We will discuss both types here. Let us start with continuous buffer system. Now, in continuous system a single buffer or constant pH is used in gel as well as electrode reservoirs. So, it is a single buffer all over. Now, sample is loaded directly onto the gel where separation will occur and they are fractionated as per their mobility. So, the height or we can say the amount of the sample volume which you are taking will determine the bandwidths, and this will limit resolution attained by the continuous system. So, what will happen if you have dilute samples, then you have to load larger amounts or larger volumes of the sample for detection of that particular molecule. And what will happen is this will certainly lead to relatively you can say broader or wide bands. So, therefore, the continuous system with constant pore size gels is kind of restricted to only high concentration, because if you apply sample in high concentration, then you get better results, not so broader or wide bands. But in dilute solutions, since you are loading too much of solutions, the bandwidths will be higher actually. So, this system has a certain problem. Let me show you on a screen how this affects the resolution as such. Alright, so if you can pay attention on your screen here. So, when we are loading the samples, as if you could recall, I was telling you about the wells or slots are created for loading the samples, and if you have a sample, if you have a gel apparatus or slab gel electrophoresis you are doing, you have a plate and you have created two plates between which through spacers you cast your gel, and what you do is in the top of the gel you will create the wells where you are going to load the sample. Now, samples could be loaded in two ways. One, like dilute solutions will be loaded here all over. Now, if I just kind of show you this here, that if I have a sample here that is at this place and this place and this place, let me show it here. Say this is your well where we are loading this sample here. Now, you have a sample here, you have a sample here, and you have a sample here, and this is the boundary or this is the interface here where from the sample is going to enter the gel. Now, obviously, when you have dilute solutions, like this is your amount of sample you have taken and it is a large volume, then since your analyte or your molecule is distributed all over, the molecule which is closest to the gel interface will enter first here and will also move somewhat here on application of electric field. But this molecule with the same analyte will there will be a time gap when it enters as relative to this one molecule, that is if I say number 1 and this is number 2, then this will move somewhere, then the 2 will enter in here. So, what will happen? This band maybe they catch up slowly later on, but still they will not be able to be like this, rather they will appear to be a broader band will be seen rather than this kind of band. So, what will happen is that with dilute solutions it is not running a continuous system is not advisable, but it has been seen for nucleic acids it does not really make so much of difference. So, for nucleic acids continuous systems are mostly run, but for proteins you get much broader band if it is a dilute solution, and so you will run rather a discontinuous system than a continuous system. So, continuous systems are not advisable in the case of proteins. Alright. So, now number 2 buffer system which we were discussing was discontinuous, or we can call it multiphasic buffer system. Now, this system uses different buffers in the gel and electrode reservoirs. If you could recall, I have shown you that on the top and bottom there will be electrode reservoirs where electrodes are placed and you have a tank buffer or electrode buffer. And is these are designed to, main purpose of discontinuous multiphasic buffer system is to concentrate or sharpen the sample zones for high resolution separation. As I have shown you a little while back, that if you have dilute solutions and if they enter the gel at different times, the broader bands will be obtained. So, best thing is to make sure when they enter the separating gel or resolving gel, then they enter as one entity, and so it is like the running line is same for all molecules, and so you can get better resolutions during the separation. So, to concentrate the sample in a very narrow zone, a large pore stacking gel which serves as an anti-convective medium here. When you say large pore, then proteins will be moving unhindered, there will not be any kind of restrictive migration here, as large pore size will not have that. So, it is only anti-convective convective medium. Now, all ionic species for moving fronts here, where buffer ion front leads. So, you have what are ionic species here? Ionic species will be certainly when you have a multiphasic buffer system, one ionic species is your protein, and other ionic species is like buffer, which is gel buffer, and then the electrode buffer. So, these are the ionic species. So, if you say all ionic species, when electric current is applied, they move a moving front actually. So, where what happens, a buffer ion front leads as it is ionized completely and moves ahead of the sample molecule. So, buffer ion, we will see what are the buffer ions and all those details here. Electrode reservoir buffer ion fronts trails behind the sample, and we will see why it trails behind, and the sample ions are between the two fronts, that is buffer ion front and the trailing front reservoir ion front. So, what happens is this sample molecules or sample ions are sandwiched, or they are compressed between the two fronts into a very narrow sharp zone in order of their mobilities. If there is a mixture of the sample, then in order of their mobilities they will settle down. So, sample molecules stack in a very narrow region. So, what you can say that it could be a few micrometers thick and may contain a very high concentration of protein, say 100 milligram per ml or so. So, stacked samples move through the stacking gel in a very narrow zones regardless of their initial sample volume, and then when they enter resolving gel, now resolving gel contains small pore size. So, what happens, the stacking these stacked samples will be unstacked and they will move according to the size and charge in the resolving gel. So, what discontinuous system is essentially doing, it is putting two gel systems, where stacking gel is to concentrate the sample, so that you can get good separation, good resolution here. So, discontinuous gel electrophoresis was like it was first developed by Ornstein and Davis in 64, and they were first to develop a high resolution polyacrylamide gel electrophoresis for the native proteins. Now, it was discontinuous and non denaturing. So, there was no denaturing agents in this gel electrophoresis system, and this is the extensively used technique in gel electrophoresis with certain modification. We will see the modifications here. Now, their system is made of 4 interrelated components here, and which includes a stacking gel, and again stacking gel will contain a buffer, a resolving or separating gel with a buffer concentrations, and an electrode buffer, that is a tris glycine buffer, and a sample buffer. Sample will also be put in a like it could be simple like stacking gel buffer, or it will have particular kind of concentrations. Now, stacking gel has a lower acrylamide concentration, like I said, it is a large pore size, it is around 4% T, and is cast on top of the resolving gel. Now, resolving gel can range from say 5 to 20% or even up to 30% you can make, but it is up to 20% mostly is utilized. Now, stacking has a lower pH and ionic strength. So, stacking gel has tris Cl, which is pH 6.8, and it has a lower ionic strength also. Then the separating gel, which has a pH of 8.8 and it has a higher ionic strength, around 0.375 molar tris Cl as compared to 0.125 molar in stacking buffer. Now, the pH difference in two gels regulate the mobility of glycinate ions from cathode reservoir chamber, and the concentration of buffers are also derived from electrochemical considerations. So, the main aim is to concentrate the sample actually, and accordingly these pH and concentrations have been derived or designed, you can say. The stacking gel with large pore size mainly functions as anti-convective support, as we were saying, and helps in formation of sharp zones without impeding the migration of proteins. Separation of proteins will take place in the separating buffer. So, remember, stacking gel is only for concentration and not really for separation here. Now, the sample buffer is little have less ionic strength, again it is a 0.0625 molar tris Cl in Ornstein Davis design, and pH is same 6.8 as that of stacking gel or stacking buffer. Now, this is loaded on the wells as I have shown you present in the stacking gel. So, sample is loaded or located between the stacking gel and the electrode buffer. Now, electrode buffer the is 0.25 molar tris and 0.192 molar glycine, and pH automatically comes to 8.3. So, the ionic strength of sample solution is lower than the buffers above and below. All right. So, let me show you on the screen like what is the situation here. So, what you have is on this gel you have two gels actually. So, what is this? This is stacking gel, this is resolving gel. So, automatically when you are casting the gel, first you have to pour resolving gel, and then after resolving gel is polymerized, then you put stacking gel actually. These are wells where the sample is loaded. So, your sample will be loaded in these wells as per like how many samples you have, and on top of this there will be, you can say, there will be electrode buffer, and top and bottom there will be electrode buffer. This is cathode chamber and this is anode chamber. All right. So, this is how the whole thing is in here. So, you can see the sample is between the stacking gel and the electrode buffer here. All right. So, let us move on. So, when power is applied, what will happen is a voltage drop develops across the sample, and it helps in driving the sample into the gel, that is stacking gel. Now, as the current begins to flow, like I said, the buffer ions leads actually. So, here chloride ions chloride ions will lead, and the gel as the current begins to flow, these chloride ions will be first to move, then proteins in the sample with negative charge will move, and then glycinate ions in the electrode buffer starts moving towards anode. So, three things which are moving in here is, one is the chloride ions, second is the chloride ions in the gel, then protein in the sample with negative charge. Remember, positive charge protein will not move towards the anode, and glycinate ions in the electrode buffer moves towards anode. So, what is the pattern here? So, all cations which I am saying tris or say proteins with positive charge will move towards cathode. So, we are it's a hard thing to separate. So, it's like you have to change buffer conditions or certain things, but we are talking about here discontinuous system. So, here you will not be able to see cationic proteins as such. Now, chloride ion with higher mobility move out of the sample, and a localized low conductivity and high field region is created behind. So, which will accelerate the proteins in the sample and the glycinate ions from the electrode buffer to same velocity as the chloride ion. Now, the effective mobility of glycinate ions is less than those of chloride ions and proteins, as the glycinate ions are not completely ionized at lower pH. Remember, pH of the stacking gel is 6.8, and even in this reservoir, electrode reservoir, it is 8.3 around. So, glycinate ions are not fully ionized, so they move like little slower. So, they trail behind actually. So, what a moving boundary is formed here with chloride ions in the front and the glycinate ions in the rear, with protein sandwiched between them. So, it is compressed between the two fronts, one is leading front, another is trailing front. Now, the protein molecules concentrate here as individual thin zones in order of their decreasing mobility. Say, if you have more than one protein, one kind of protein or one type of protein molecule, then different protein molecules as per their mobility, they will concentrate as thin zones, but they will concentrate here between two fronts, leading and trailing glycinate ions. Now, when the moving boundary region will reach the interface of stacking and resolving gel. So, in a stacking gel, what is the situation? That due to these pH differences, ionic differences, it is like one front, that is chloride front, leads and the glycinate ion front trails, and it will in between proteins are sandwiched and they are compressed, and finally, sharp zones are formed, and this particular process is known as isotachophoresis. So, when these are concentrated and they reach the interface of stacking and resolving gel, so they will concentrate at that interface because they are going as thin zones here, and they will enter into the resolving gel as one entity. So, as they reach there, the chloride ions and the glycinate ions which leaves behind the protein. Now, here if you remember, the pH of resolving gel is 8.8, and as the glycinate ions move, the pH is much higher of the resolving gel, and so the glycinate ions are not fully ionized and they move as per their velocities. But proteins, because now it is entering a low pore size gel, so there will be a restrictive migration as per their size and charge, and so protein will now they will enter the resolving gel and they will experience a sharp retardation due to the resolving gel, smaller pore size of the resolving gel, and these protein zones which we were talking about, which were stacked actually, that is why it is called stacking gel. Now, they unstack, and since they are of there was no hindrance or movement in stacking gel, but here since small, large or other proteins will have to move according to the pore size. So, they will unstack and separate according to their size in charge. So, what you see is that all these factors will result in the proteins becoming compressed at the interface of the gels, and therefore, giving you very high resolution in these experiments. So, this is discontinuous system is quite useful system for higher resolution for protein separation. Now, this figure shows the things very clearly here. What you see here, like I showed you, there is a tank buffer which is cathode electrode is there, negative electrode. There is a tank buffer tank below, reservoir below, which is also filled with reservoir buffer. So, there is reservoir buffer here and here. There is a separating gel which is larger. If you can see, most of the plate is covered with separating gel, and there is a very small part is stacking gel here. You load your samples in this wells, and if you can see, these samples are distributed here, big, small, but they form boundaries here, thin zones, and when these thin zones enter, like the one here, when they enter the gel at the interface into the resolving gel, they will separate according to their size and charge. So, this is how discontinuous system works actually, and it is very well depicted in here. All right. So, this was about two buffer systems which are widely used, one continuous system and another is discontinuous or multiphasic systems. Alright, so we will start with the electrophoresis of proteins. Now, there are many variations of electrophoresis which are commonly used. There are a lot of techniques which are used for different applications for analysis of proteins, and we will discuss some of them. Like there are native gel electrophoresis. Now, native gel electrophoresis is one where analyte separated is according to the differences in apparent mobility, and protein is not denatured here. Then there is SDS-PAGE or sodium dodecyl sulphate polyacrylamide gel electrophoresis, where what you see is that analytes are denatured because of SDS and they are separated according to size. And then there is isoelectric focusing, where in isoelectric focusing the separation is on the basis of the pi values of proteins, as the proteins are amphoteric molecules and they carry a net charge. The charge is 0, net charge is 0 at their isoelectric points, and that becomes the basis for separation. And then there is two dimensional gel electrophoresis, which is quite used in with mass spectrometry techniques, where you can separate large number of protein samples in a particular proteome. Say you are isolating it from certain cells or tissues, and then they could be analyzed by mass spectrometry. So, we are going to discuss all these techniques. Today we will be discussing about native gels and SDS-PAGE. So, native gels, as we were discussing in the continuous and discontinuous system, like Ornstein and Davis system was for native proteins actually, where you have not put any denaturing agent actually. So, in native gels, non-denaturing conditions are used to detect the protein in its active form, particularly in case of enzymes, where you would like to see the activity of the enzymes, then you might want to use the native gels. Now, in native gels, polyacrylamide gels are used, but the SDS or any other denaturing agent is absent. So, you do not denature the protein in any way here. Since all the proteins in the samples being analyzed carry their native charge at the pH of the gel, proteins will separate according to their different electrophoretic mobilities and the sieving effect of the gel. And various types of native gels, like you can use, there is no restriction on the pH of the buffer. You can very well design, or you can as per your requirement you can use certain buffers for native gels. Then you can detect it by various methods, which we are going to discuss in later lectures. So, this was about SDS-PAGE actually, this was about native gels. Now, one of the main techniques which is widely used in a lot of different research labs and in teaching labs is the SDS-PAGE or sodium dodecyl polyacrylamide gel electrophoresis. So, we were discussing about Ornstein and Davis discontinuous and non-denaturing system, if you could recall a while ago. Now, this particular system has been modified by Lamelli to a discontinuous, but denaturing system. Now, Lamelli system made it possible to determine the molecular weight or molecular mass of the proteins. How it is done, we will be discussing that. So, what happens here, rather than having like in Ornstein Davis, where no denaturing agent was used, in this system of Lamelli there is a denaturing agent, which is 0.1% SDS, that is an anionic detergent, will be utilized in buffers. And also it will be introduced like one more step was introduced, that is denaturing step in sample preparation or sample treatment. So, what happens, if you treat the samples which contains around 2% SDS, and also a heating step is introduced here, which will completely denature the sample proteins to their constituent polypeptides before the run. So, here sample treatment involves like I said, heating in particular buffer, and this buffer, which is like mostly stacking gel buffer like I told you earlier, and but this buffer with sample buffer which we were discussing in Ornstein and Davis system will also be having 2% SDS and also a reducing agent like 5% beta mercaptoethanol. So, so the proteins are completely denatured and polypeptides will take a uniform charge to mass ratio which is imparted by SDS. Also reducing agent will break any disulfide bonds between polypeptides or within polypeptide chains. So that SDS can be interacting with all parts of the polypeptide chain. Many times what happens when disulfide bridges are not broken, there are regions where SDS is not accessible and maybe erratic patterns could be seen in here. So, polyacrylamide gel electrophoresis in the presence of SDS is the most common form of protein gel electrophoresis. Now, here there is lithium dodecyl sulphate, that is an alternative which could be utilized, but SDS is the most widely used detergent. Now, SDS disrupts some of the non-covalent interactions that stabilizes the protein quaternary and tertiary structures. And facilitates denaturation. What happens? SDS has a negative charge, and what it will bind to the protein in a constant mass ratio, that is mass ratio of 1.4 gram of SDS binding to 1 gram of polypeptide. So that total amount of detergent bound is directly proportional to the molecular weight of the protein. So, it is 1.4 gram of SDS per gram of polypeptide or protein, you can say. So, what it does is SDS coats the polypeptide completely, and coating of negatively charged SDS overwhelms the inherent charge of protein molecule and gives them a uniform charge to mass ratio. So, this is like now protein will carry a net negative charge as per its molecular mass because of the interaction of SDS. Now, remember many times though this happens, but certain proteins with say a lot of basic residues say lysines or others there might be certain discrepancies in charge to mass ratio, and there might be certain problems in running, but in general this is the rule where you have a particular mass to charge ratio actually. So, so this allows proteins to be separated on the basis of their relative size, because the charge, that is negative charge, is according to their molecular mass. So, you are essentially separating it on the basis of size actually. So, what happens is as we were discussing about what happens to proteins. So, a protein is folded here, it has interactions and so there are hydrophobic areas, there are other charged groups, hydrogen bonding, all those things. Now, when you treat it with the SDS, protein is extended and it is overall covered by the negative charge. So, that is what now many times these proteins which are have different shapes also, they are now extended molecules, and heating also helps in that. Now, many times also you can use other denaturing agents. For example, urea is quite commonly used denaturing agent which disrupts the hydrogen bonds, and it is widely used for nucleic acids as we will discuss later on, but for proteins also it can be used, but SDS is the most widely heating, and SDS is the most widely used method. Now, advantages here of this particular method is that all polypeptide chains are now forced into extended conformations because of SDS. SDS treatment eliminates the effect of differences in shape also, like I was saying that shapes are now all extended. Then individual polypeptide chains migrate as a negatively charged SDS protein complex through the porous polyacrylamide gel, and the speed of migration is proportional to the size of the proteins. Smaller polypeptides will run faster because they will have less restrictive or hindrances, and larger polypeptides will run slower because the pore size will be smaller and they will be kind of resistance will be higher for larger polypeptides. So, these are you can say the advantages which SDS provides, SDS treatment provides for running the electrophoresis of proteins in denaturing conditions. Now, if you can see in here figure, you have two like samples here. One protein, if you can see here, is having disulfide linkages, one is without disulfide. Now, when they are heated in presence of mercaptoethanol, these disulfide links are broken. It is a reducing agent, so it reduces that. And then if when a single protein, though it is made of two polypeptide chains, but it will be seen as two separate bands here in reducing gel. Now, to know whether it is a single protein, always you can run non-reducing gels also. In non-reducing gels, these disulfide bridges will not be broken, as mercaptoethanol will not be included in the sample buffer, and single band could be obtained in here. So, both ways you can, like say if your protein has say a certain disulfide bridges between the polypeptide chains, then if you are utilizing reducing gel, certainly you will get two bands, you will not be able to know whether it is pure or not if you are testing the purity. So, then you must run a non-reducing gel, that is sample buffer not having mercaptoethanol, and if single band comes, then you will be sure that it is a same protein, it is a single protein made of two polypeptide or more polypeptide chains. So, how this SDS-PAGE is performed, the practical aspects of this. Now, the first step in discontinuous electrophoresis is to pour the separating gel, like I told you, and in earlier lecture I have shown you how to cast the gel. So, there what you are going to do, first thing is resolving or separating gel will be casted first, then separating gels will typically contain like I said 6 to 20% of acrylamide, and the size range of the proteins being separated has to be taken into account, desired resolution, amount of sample being applied, all these factors needs to be considered while choosing the acrylamide concentration. Once you have casted the resolving gel, then once it is polymerized, you will put the stacking gel and with comb in it, like I have shown you earlier, so that wells could be created. Now, there could be resolving gel where you can use gradient gels also, and these gradient gels, it is a pore size gradient gels we can say, because there will be either decrease in the pore size as you move down the glass plate. So, that will give better separation. It could be step gradient or it is a continuous gradient gel, and in many situations where you have to examine both high and low molecular weight proteins on the same gel, the gradient gels are more advantageous. The stacking gel is poured after the separating gel polymerizes and just before electrophoresis is done. Once these are polymerized, and you have to take care that both resolving gel, when it is polymerized, you can overlay it with say organic solvent or so that surface labels properly, and then you can remove that and then pour the stacking gel and put the comb. Now, once it is done, you load your samples into the wells of the gel and the current will be applied. Now, samples for SDS-PAGE are prepared in sample buffer as we have discussed earlier, that is 0.0625 molar tris, pH 6.8, but it contains 2% SDS and 5% mercaptoethanol if it is a reducing gel. Also 10% glycerol is present. Now, mercaptoethanol is added just before the experiment, and glycerol will provide density for underlaying the sample on the stacking gel below the electrode buffer. So, like since buffer is already put in, if you have glycerol, it will not diffuse and properly settle down in the well. A tracking dye that is bromophenol blue around 0.025% is also included in the sample, and this dye reaches the bottom of the gel, and so what you will know is when to turn off the power, and that is the like fastest. So, you do not want to go after that, otherwise your samples might overrun. A common way to detect proteins after electrophoresis is performed is to stain the gel, that we will be discussing later on. Before staining, gels are usually fixed with acetic acid methanol solutions and like 10% acetic acid, 40% methanol or so, and which what happens is it fixes the protein or precipitates the protein into the acrylamide matrix. So, they do not move or diffuse, and you can easily do the staining. After staining, the gel will look something like this. You have like here, these are molecular weight markers, these are different samples which have been done, and you can see these blue bands here, which has been stained with Kamasi brilliant blue, and this is you can get a very good this is how you will you can see your gel and different band patterns. Now, like I was saying, this gel here which I have shown you, mostly it is for the high molecular mass proteins. When you have to run very small molecular mass proteins, then it is better to have rather than glycine buffer, you can have trypsin buffer. Trypsin buffer is better for smaller polypeptide chains, and they will stack they will separate better in trypsin buffer. SDS-PAGE is widely used for, like we say, calculation of molecular mass. So, you can identify the protein and get some information about it. Now, molecular masses of proteins can be estimated by comparing the migration of proteins of interest to standards of known size. If you could recall, like I told you earlier, that gel is a very random meshwork, like it precipitates and fibers are formed and then they form a random structure. Many times these things cannot be repeated actually, as every time you cast a gel it might be different. So, you have to really compare it with standards actually every time you run your sample. So, the relative mobilities of the standards are plotted against the log of their molecular masses, and then you can compare the size of the unknown protein and then extrapolate from the standard curve. So, here what you do is a relative mobility or RF value is calculated. If you can recall, when we were talking about the chromatographic techniques, particularly in thin layer and paper chromatography, the RF value is a very important factor. So, here also it could be like calculated by dividing the distance the protein migrates to the distance total length of the gel or the dye front actually. So, what is done? For example, this is your standards here. So, the migration of the standards could be taken. So, you have taken like these are the molecular mass here, you take you can calculate the log of molecular mass, and then here distance migrated. So, you have distance like 14.5 millimeter for this band, and likewise 92 millimeter for this band, last band, and total gel length will be around 100 millimeter. So, RF value can be calculated by dividing by the total gel length, and then you can plot the RF value log molecular mass against the RF value, and you can this standard curve can provide you the size of the unknown sample. So, this is how very easily and molecular mass it is approximate, I would say approximate molecular mass could be calculated here. So, SDS-PAGE is a very useful technique to be used. So, in this lecture, to summarize, we have discussed about the continuous system, buffer system, and the discontinuous buffer system. The continuous buffer system utilizes a single constant pH buffer all over, like in electrode buffer or gel buffer, whereas in discontinuous system there is a multiphasic buffer system, like you have say stacking gel buffer, you have a resolving gel buffer, electrode buffer, sample buffer, likewise. So, Ornstein and Davis made this for polyacrylamide gels first time, and this was a very it gave high resolution, because you can concentrate the protein in stacking gel, and then when they enter as one entity into the resolving gel. Lamelli improved it further for molecular mass determination by making it denaturing gels, and introduced he put 2% SDS, and that is the most widely used electrophoretic technique in, or routinely used technique in a lot of labs here, biochemistry labs. So, 2% SDS-PAGE in sample and 0.1% SDS in buffers, which denatures through heating, and mercaptan ethanol reduces the disulfide bridges. So, polypeptide is completely extended, and all different proteins are completely extended and overlaid with a negative charge. So, they move according to their size actually, because charge is the ratio is same, mass to charge ratio is same. So, that way you can compare with the standards proteins, and you can calculate the molecular mass of an unknown sample or unknown protein here. Also, SDS-PAGE gives you a clue about the purity of the sample, whether say you have gone through a purification procedure, and then first thing to do is whether your purification is done or not, you have accomplished the purification, the SDS-PAGE is run to see that you get a single band. Now, remember, even if you get a single band, it is not 100% purification. There might be impurities which are not seen because you are utilizing a particular kind of detection system, like say Kamasi brilliant blue can give you to a certain extent, as we will discuss later on, but silver staining can further give you is much more sensitive and can tell you about purity if you want to know so. So, in the next lecture we are going to discuss about two more techniques, that is isoelectrofocusing, where PI is taken for separation, and 2D gel electrophoresis, which is like to separate many samples in 2 dimensions, that we are going to discuss in next lecture. Also, we will be discussing about detection methods of proteins and preparative gel electrophoresis in the next lecture. Thank you.